And welcome to the section of Chapter 5 in the Campbell Biology textbook on proteins. Remember, just a reminder, we're talking about macromolecules here. Remember, there's 4 major macromolecules that make up living systems. Remember, macromolecules are larger molecules that are a lot more complex than, let's say, more simple molecules that you've learned at previously. So remember, the 4 classes of macromolecules are going to be carbohydrates, lipids, proteins, and nucleic acids. This section focuses on proteins. Remember, each one of these 4 classes of biological macromolecules have unique properties that arise from the orderly arrangement of their atoms and the different combinations of functional groups that are attached to their base skeletons. So proteins can create a great diversity of structure. They have a wide range of functions as well. In fact, proteins are quite important for the cell. They make up about 50% of the dry mass of most cells. So what does that mean? If you took all the water out of a cell, 50% of the molecules inside that are going to be proteins. So they're pretty important. They have a lot of different functions. One of them is enzymatic. So some of them act like enzymes. And what enzymes do is catalyze chemical reactions. So they help speed up chemical reactions. So here's an example, right? So digestive enzymes are ones that actually catalyze the hydrolysis of molecules that can provide energy, such as perhaps sucrose. Remember, we talked about sucrose, or you saw the structure of sucrose and the formation of sucrose in one of the other segments. Now we're talking about the hydrolysis of sucrose to extract that energy. Cell proteins serve as a function in defense against foreign bodies. So antibodies, you've probably heard about them. Antibodies are actually proteins, and what they do is try to inactivate or stop viruses and bacteria or other pathogenic materials from causing disease in the body. Antibodies are often produced by your body in response to an infection. Some of them store energy. So they can store energy in the form of amino acids. So one of the monomer of proteins are amino acids, and amino acids can carry energy. Casein, for example, is a milk protein. It's actually a major source of energy in the form of amino acids for baby mammals and eggs. Plants have storage proteins in their seeds. Ovalbumin is going to be a protein in egg white that is used as a source of energy for a developing embryo. So ovalbumin is the protein. It's contained in the eggs, and it's made up of amino acids, and these amino acids will be used by the growing organism as an energy source. Some proteins serve to transport things inside and outside of the cell, or across the cell membrane. Hemoglobin is an example that doesn't transport across the cell membrane, but it does transport oxygen to different parts of your body. And it takes carbon dioxide away from parts of your body. So you take in oxygen and you breathe out carbon dioxide. Hemoglobin has an iron component within it, which actually attaches to oxygen. So it kind of allows hemoglobin to carry oxygen, and it carries oxygen from the lungs when you breathe in, and it delivers the oxygen to the extremities, to the capillaries within your body. At the capillaries, when it releases that oxygen, it grabs onto the carbon dioxide that's generated during cellular respiration and delivers that carbon dioxide to your lungs for you to exhale. Some proteins serve as hormones. Remember, we talked about steroid hormones, but there's also protein hormones as well. And hormones are signaling molecules which signal the body to either produce something or do something. Insulin is an example. You've probably heard of insulin before. And you may be diabetic, or you might know somebody who is diabetic. So insulin is actually a protein, and it's a hormone protein that's secreted by the pancreas. And it's secreted by the pancreas in response to high blood sugar. So it signals the body to store that sugar, or do something with that sugar to remove the blood, or the high amounts of sugar in the blood. Some proteins serve as receptors. So receptors are typically on the surface of cells, and receptors have a specific shape, and they're meant to bind to specific molecules, or some specific molecules are meant to attach to them in some way. And what receptors do is, once the molecule that they're specific for binds to it, it sends a signal from outside of the cell to the inside of the cell to the cell to do something. In the nerve system, right? So we have a nerve cell right here. We have signaling molecules that can occur between two different types of cells. And this particular molecule, signaling molecule, is going to be released by this cell. And then it's going to bind to the protein on the surface of another cell from perhaps a different tissue type, and then induce some sort of chemical reaction inside those cells. Some proteins serve as a function in contraction, especially in muscle. And some of them are motor proteins. Motor proteins are interesting, and they have an interesting shape in that they have almost like legs and feet, and they walk along other proteins. So basically this allows movement within the cell or between tissues. So here's an example of motor proteins and other proteins that are involved in muscle contraction. So we have actin, which is one type of protein, and myosin, which is another type of protein, and they interact with each other to allow contraction of muscle tissue. And then some of them are just structural proteins, so they just provide some sort of support. One example would be keratin. This is the protein that's found in hair and feathers and other appendages on the skin. Insects and spiders use silk fibers to make their cocoons and webs, respectively, so silk fibers are a type of protein. Collagen and elastin are proteins that are found within the connective tissue in animals. So here we can see an image of connective tissue, and within that connective tissue we have a collagen molecule. Now, a protein, another name for a protein is going to be a polypeptide. And these are the polymers. So a protein is the polymer, and the monomer is actually amino acids. So when amino acids connect together, they create a polymer, and the polymer can be called a polypeptide. And the reason why it's called a polypeptide, not really a polyprotein or polyamino acid, it's called a polypeptide because when amino acids connect together by that chemical reaction, dehydration synthesis, it creates a peptide bond. So protein is basically it's a functional molecule, and sometimes it is one polypeptide, which is one strand. Sometimes it's made up of more than one polypeptide. So let's talk about the amino acids, which are the monomers of proteins, or polypeptides. So amino acids are, of course, organic molecules. They are made up of carbon, and they have both or two functional groups. So every single amino acid has at least one amino group, which is shown here on the left, and one carboxyl group, which is shown here on the right. In between the amino group and the carboxyl group is a carbon. This carbon is called the alpha carbon. The alpha carbon has, of course, the amino group and the carboxyl group attached to it, but also a side chain called the R group. This side chain differs between each amino acid and can consist of or be another amino group. It could consist of also an amino and another carboxyl group, or just another carboxyl group, or just consist of a carboxyl group. So we'll see an example of this in just a moment. So again, this is the overall structure of amino acids, and it's important to note that every single amino acid has at least one amino group and one carboxyl group, but there could be one or more of those in the side chain. Now let's look at a group of amino acids. Now there's 20 total, And here we can see there are 9. There are 9 amino acids that are considered hydrophobic. So what this means is that their side chain is considered hydrophobic. Now it's important to note that the part that all amino acids have is the amino group and the carboxyl group. This makes this part of the molecule hydrophilic because the amino group is typically charged and so is the carboxyl group. So they're basically opposite charges. And here we have basically, these are amphipathic or amphiphilic amino acids because they have hydrophobic regions and hydrophilic regions, similar to the fatty acids that we talked about. However, these more readily dissolve in water. However, the ones that are larger, in terms of larger hydrophobic groups, those are going to less readily dissolve in water because they have more hydrophobicity to them. So you can see all the side chains that are hydrophobic either have just carbon and hydrogen or just hydrogen. So we have carbon and hydrogen, carbon and hydrogen. Now we do have a nitrogen here, but it's involved in a ring system, so it doesn't really create any electronegativity. And we have a sulfur here, again, not very electronegative because there are 2 carbons and methyl group attached to it as well. So that's 9 of them. And it's important to note that each of them have a name. So glycine, alanine, valine, those are the full names, but then they have other abbreviated forms of their names. So glycine here is GLY, that's the 3-letter abbreviation, and then there's a single letter abbreviation. For glycine, it's G. Now, not all of them are going to start with the same letter or very similar to the same word. Here we have tryptophan, TRP, however, the single letter is a W. Phenylalanine, PHE, but then the single letter is F. And you'll see some significant variations in just a moment. So those are the hydrophobic ones. Now let's look at the hydrophilic ones. And of course, as opposed to the hydrophilic ones have polar side chains. So these are side chains that have electronegative atoms. So here, most of them have oxygen, but there are some that have nitrogen. And the nitrogen here for these are going to be on the end. So when it's on the end, it's more exposed. So it's going to create more polarity. Same here with the sulfur. Right, so this is hydrophilic. It does create more polarity since there's not a carbon on the other side, so it's just a hydrogen attached to it. So there's going to be a difference in electronegativities on either side of the sulfur. Okay, so these are the hydrophilic ones. So here we have glutamine, GLN, and then the single letter code is a Q. Now we have those that are charged. So just like the amino group and the carboxyl group that every single amino acid has, these have another either amino group or a carboxyl group in their side chain. So those that are negatively charged are going to have another carboxyl group, and those that are positively charged are going to have another amino group. So the negatively charged ones act more like acids, and then the positively charged ones act more like bases. Now, polypeptides is a term that is used to describe a protein, right? So we've already kind of talked about this. It's a polymer of amino acids. Now, how do the amino acids come together? By the same dehydration synthesis reaction that we've been talking about in carbohydrates and in when fats come together, and also nucleic acids. So what happens when this occurs is a peptide bond forms, and that's why we call it a polypeptide. So here we can see that the carboxyl group on one amino acid is going to react with the amino group on a new amino acid, whereby water is going to be removed, the dehydration part, and then we create a bond between those, and this is the peptide bond. So there will be 2 peptide bonds between 3 amino acids, and there'll be 19 peptide bonds in 20 amino acids, and so on. Now, there are 20 amino acids, as you saw the structure, and they're classified based on their side chain properties. Now, there can be different combinations of amino acids connected together, similarly with the nucleic acids. This is the protein sequence, and the protein sequence can be studied in different organisms to identify various similarities and differences between proteins in different species of animals. This is another way of investigating evolution. Now, if we look at this structure, these three different structures, these are three ways in which proteins can be shown in your textbook. So you might see these various different ways. So we have the space filling model. The space filling model shows the amount of space that each atom takes up inside the protein. So it looks like a big glob of stuff. So this really shows everything of the protein. And then we have the ribbon model, which just shows the backbone and it shows the secondary structure of the protein. So here we can see an alpha helix, which we'll talk about in a little bit, and then we can see some straight chain regions, which are beta sheets. So again, this shows the backbone, and I'll talk about the backbone in just a moment. And then this wireframe model shows both the backbone, which kind of shown here as a ribbon, but then also the side chain in a stick or wire type of figure. And then here, basically, this is a target molecule. So this is basically a molecule that might be on the surface of a bacteria. And this part right here is supposed to represent lysozyme, which is actually a protein that breaks down the cell wall of bacteria. Now let's look at some simplified diagrams of various proteins and their structures and function. So sometimes there's going to be a simplified diagram which kind of shows the protein. Here it just shows it in a membrane, but it's transparent so that you can see the secondary structure and a molecule within it, or part of the protein, which might be an attachment. And then you might see a solid shape, which doesn't show as much detail, but it does show it crosses the membrane similarly to the transparent shaped one. And then you might see a more simple shape, which is basic. It just shows the enzyme as like a oval type shape binding to its molecule. So here this shows an enzyme that's actually catalyzing a chemical reaction which separates these two sugars in this molecule right here. And then you might see a protein shown as little balls. Right, so here we can see insulin. Insulin is a protein. Remember, it's an example of a hormone protein, and it's shown as just little balls. All right, so let's go ahead and talk about the sequence of amino acids of proteins. So the sequence of amino acids determines the three-dimensional structure. So depending on what order the amino acids are put together really directs what the overall three-dimensional shape of the protein is going to look like. So a protein structure, its three-dimensional shape, really determines kind of how it works. So if a protein hasn't folded into its native, it's called native, its preferred shape, then it's likely not going to function properly or function at all. So the function of a protein usually depends on its ability to recognize, or sometimes recognize another molecule and actually bind to it. So here's an interesting example. So we see just the backbone, and this is in the wire diagram for two different proteins. One of them, the one on the left, is an antibody, and the one on the right happens to be a protein from a flu virus. So the antibody happens to be produced, and it looks like it is complementary. So it fits right in there, almost like a lock and key. So it fits right in there, and it allows it to bind and stay bound to it. So once that antibody, if the antibody can bind to that surface of the protein, so this just shows the surface of, let's say, if you had the space filling model, but this shows the surface of it. And it shows how the surface is complementary between the two and how antibodies can actually bind to proteins from, let's say, a virus or a bacteria and actually stop it from doing whatever it's going to do. So that's how antibodies protect us from disease. Now there are 4 levels of protein structure. So we're going to discuss each one of those levels. The primary structure. What stabilizes the primary structure? The strongest bonds possible. Well, not the strongest bonds out of the major types of bonds, which is going to be covalent bonds. So the peptide bonds that occur between each amino acid is a covalent bond, and those are very strong. So it's really hard to break up amino acids, or to break up the bonds that between amino acids. Now it is a little bit easier to break the secondary structure because it's just hydrogen bonding. Hydrogen bonding is really a weak bond by itself, but many hydrogen bonds can make things very strong. Now, if we look at the tertiary structure, this is still one peptide or one chain. This is when primary structure is made, and then the secondary structure starts to form, and then the secondary structure starts to fold upon itself, and it creates this three-dimensional structure. So here we can see an alpha helix and beta pleated sheets within the whole tertiary structure and the transthyretin. Now, not all proteins have quaternary structures. So far we've talked about the primary, the secondary, and the tertiary. All proteins have primary, secondary, and tertiary structure. However, some proteins have quaternary structure, and the quaternary structure is formed when more than one peptide chain comes together. Now, as I mentioned, the primary structure is stabilized by covalent bonds. The secondary structure is stabilized by hydrogen bonds. Tertiary structure is stabilized by all different combinations of bonds. They could be covalent bonds, they could be hydrogen bonds, they could be van der Waals interactions or hydrophobic interactions, or ionic bonds. So if we look at this, this is an example. This just kind of shows the ribbon, which would represent the tertiary structure of the protein. And again, there can be hydrogen bonds, hydrophobic interactions or van der Waals interactions, disulfide bonds, which occur between cysteines. Now, this is a covalent bond, and then, of course, ionic bonds. And it could be different combinations of these that occur. And it's going to occur within the strand because, again, we're talking about tertiary structure. This is only one single strand here. Now, these are hydrogen bonds here. It's not like the hydrogen bonds in the secondary structure. The secondary structure, the hydrogen bond occurs between the atoms that are involved in the backbone, which are those atoms involved in the amino group and the carboxyl group in every amino acid. So that's the backbone part. But there could be amino groups, carboxyl groups, hydroxyl groups, other types of groups that can interact with each other in the side chains. So it's side chain interactions that stabilize the tertiary structure. Now, quaternary structure, like I said, this involves more than one peptide. So not all proteins have more than one peptide. If a protein does have more than one peptide, they're each called subunits. And they're each called subunits because typically the protein isn't going to be completely functional until all the subunits are put together. So here we have the full transthyretin protein. Again, we were just looking at one of the subunits up to the tertiary structure. And this protein happens to have quaternary structure. And you can see there are 4 subunits here. 2 of them are the same, and the other 2 are the same type of structure. Now, what stabilizes the quaternary structure are the same interactions that can stabilize tertiary structure. It's side chain interactions. So these side chain interactions can involve ionic bonds, they can involve hydrophobic interactions or van der Waals, they could involve hydrogen bonding, and even covalent bonds, usually between cysteine amino acids. Now, this again just kind of represents that. Remember, we just saw this picture. These are the same types of bonds that can stabilize quaternary structure and bring together and keep together subunits within a protein. Now, if we look at some examples, this is an example of collagen. So collagen has quaternary structure because there are 3 strands that are kind of looped together like a rope. So they're kind of tied together like a rope, and each strand has secondary structure as well, and tertiary because it kind of folds, kind of wraps around itself, really. And the secondary structure you can see definitely alpha helices. And of course, all proteins have primary structure. This is another example. This is hemoglobin. Hemoglobin has 4 subunits, so it has quaternary structure. It also has some attachments to it. So it has this heme group, which actually incorporates the iron. So that's where the iron is located. And it's the iron that actually binds to oxygen, which allows the transport of oxygen to your extremities. And then, of course, once it releases that oxygen, it can actually grab onto carbon dioxide and then bring it back to your lungs so you can exhale the carbon dioxide that's generated during cellular respiration. Hemoglobin is going to be found in your red blood cells. So it's going to be found in your blood. And again, your blood kind of reaches all your extremities, and that's how you get oxygen in your entire body. So protein structure and function can be affected by a number of things. So a change in the primary structure, which would be considered a mutation, can affect the protein structure and eventually its function as well. So a key example, or a classic example, is the change that occurs in sickle cell disease in those individuals that have sickle cell disease. Back to this, we have amino acid number 6. This is a glutamate. So glutamate is actually a charged amino acid. It's negatively charged. But in sickle cell, it's changed. The DNA sequence has changed so that it during protein synthesis, a valine is incorporated instead of glutamate. Valine is a hydrophobic amino acid, so it's got different properties than the one that should be there. And again, remember, this affects the sickle cell subunit, and it creates a different shape in the beta subunit. And this creates this shape where you have this valine sticking out on the surface. And again, it's hydrophobic. So remember, in the cell, it's mostly water, and hydrophobic things don't like water. Remember, they're hydrophobic. So what happens is this hydrophobic portion that's exposed in the beta subunit, once the quaternary structure is formed, starts to aggregate with other hemoglobin molecules that also have that structure. So they're basically they're trying to hide that valine, or the valine doesn't like water, so it kind of aggregates. So remember, I was talking to you about how the membrane can self-assemble due to the hydrophobic and hydrophilic properties of phospholipids. Well, this is kind of the same idea. So remember, the phospholipids are kind of embedded in a double membrane to hide the phospholipids, or the hydrocarbon portion of the phospholipids from water. Well, this is basically aggregating so that the hydrophilic portion is exposed to water, but the hydrophobic, what's sticking out, this mutation, is kind of trying to stay away from water. And this creates these long fibrils inside the blood cells, which then affect hemoglobin's ability to deliver, or the red blood cells' ability to deliver the oxygen from this hemoglobin, right, to the extremities. These blood cells start to look like a sickle shape, and they get stuck in the extremities, so they do not deliver oxygen very well, and this can be fatal. However, there are for this. So this shows the difference between a normal red blood cell on the left, so it kind of looks like a smooth donut, and then a sickle cell blood cell on the right. So you can see it's got the sickle shape where it's got these fibrils that are making this shape. Inside the normal one, we've got hemoglobin just kind of floating around in there, and these red blood cells in the blood. Now, other things can affect protein structure and determine what the protein structure is. So in addition to changes in the primary structure, physical and chemical conditions can affect the structure and ultimately the function of a protein. So most proteins have a preferred pH in which they work best. Some of them have a preferred salt concentration, some of them preferred temperature, and sometimes there's other environmental factors that can be at play. Now, if there's any change in pH or salt concentration or temperature, then this affects the function of the protein and sometimes the structure of the protein. So proteins, they kind of fold, and let's say this is the normal ribbon diagram for a protein in its native structure. So if we go across the top here, okay, where it says denaturation. So when the conditions in which a normal protein change, and the protein doesn't like it, it can affect the structure of the protein, and it can what's called denaturation, but it's like unraveling of the protein. So it doesn't affect the primary structure. So we have the amino acids still connected together, but the secondary structure, tertiary structure, and quaternary structure, if it has any, is affected. Now we can take a denatured protein and renature it very slowly so that it can fold properly in its native form or functional form. So denaturation is the term in which what happens to proteins where they kind of unravel due to undesired environmental conditions. Now protein in the cell, so it's difficult to predict a protein structure based on its amino acid sequence, although there is software online which kind of can give you some predictions. But it typically, and it most likely goes through several stages of folding on the way to the most stable structure. So it might, of course, it's going to start unfolded, but then it's going to start folding a little bit and then maybe change its shape and then ultimately fold into its most stable structure. And its most stable structure is likely the structure in which it's most active or it prefers. Now scientists use X-ray crystallography to determine a protein structure. So what this is, we have this X-ray source. When we have the beam, it goes through the crystal, so the proteins are crystallized, and then basically you have this detector that detects the diffracted X-rays. So these are X-rays, they go through the crystal and then they're diffracted, they're kind of moved into some way. And then you look at this X-ray diffraction pattern, and they can use it, I mean, with a lot of knowledge. I don't have the background for this, but they can use it to actually predict the structure of proteins. So this would be a predicted structure of the protein in which this crystal represents. Now, diseases like Alzheimer's disease, Parkinson's, mad cow disease are associated with misfolded proteins. So these are the diseases right here, and the genes that are affected. Okay, so again, each of these genes are going to produce a protein. So the gene is in the DNA, and then after the gene is expressed, we have proteins. Now, sometimes these proteins can be produced, but then they are misfolded, or they're never folded properly to begin with. When proteins are misfolded, they start to aggregate, similar to what I was showing you with the hemoglobin, because when proteins are misfolded, there's a lot of hydrophobic regions which normally aren't exposed. They're going to be exposed because it's not folded properly, and they can aggregate and they can create plaques. They're often called plaques, and this usually happens in the nervous system, and they're stored like in the nervous system, and this causes neurodegeneration in most of these types of diseases. Prion disease can be spread between different animals, including humans. So prion disease is basically the result of a prion, which happens to be a misfolded protein. So all of these misfolding can affect the organism as a whole and can be fatal. Now, I just want to kind of summarize here. The structure and function of proteins was discussed. So if you remember, the components of proteins are amino acids, and those are the monomers. And when monomers undergo dehydration synthesis with other monomers, they create polymers. And there are so many examples of proteins. Remember, if you dried out a cell, took out all the water, 50% of it would be proteins, because proteins, look at all the functions. There's so many proteins, and they're all necessary for proper cellular function. And you can see all of the various functions. So you have different examples, types of proteins, and then their corresponding functions. All right, thank you for listening.